Okay, it is Wednesday. What is it? Wednesday, 23rd? Yeah, 23rd. Okay, so Wednesday, 23rd, and we're discussing female reproduction, which is chapter 22, and we have an exam coming up on Monday. So I'll try to post all of these videos. So I've been, I had a problem with oversaturating the network, where the Dominican YouTube site won't accommodate that many lectures being posted. So I seem to have found a way to get around it, at least for a little while, so I posted a few more since then. So I'll try to fill it in a little bit more as well. So it's one easy way to study for the exam. So, okay, so we had drawn, prior to this filming, we had drawn a cartoon of what we know about the structure of the female reproductive system. We had correctly labeled uterus, cervix, vagina, and then their connection through uterine tubes of the ovary, and then there's this interesting looking finger-like projections of the fimbriae, which as part of the infundibulum, infundibulum. And we are now on to talking about the, this is not what talking about the maturation of oocytes, and that there needs to be a lot of support cells, and that just like in the male reproductive system, where support cells, there was the interstitial cells that are producing testosterone, which are significant, but even more intimately associated with the sperm-producing cells and the development of spermatids was the was the Sertoli cell. And so then we've got a similar situation with the female reproduction. I'm acting a little bit finicky today. I think maybe when I carry the disc around in the book and it rubs against the book, after a while it has these little micro scratches in it. Maybe that's what's happening. Could be. Okay, so I'll just think I'm starting it again and see what happens. Okay, so we're trying to get to figure 3. We could also look at figure 2. So there's the maturation of the follicle. So when we're talking about follicles, we're talking about the oocyte and its associated cells. And as there are more and more associated cells, you go from a primordial follicle to a primary to a secondary follicle to a vesicular follicle, and eventually to the release, the ovulation of that oocyte. And that's kind of good to look at. Let's see. So I was just pointing out that the follicular cells, once there's a certain amount of them, they're granulosa cells. But there's some other structure there too. And let's get to that. So will it let us go to the next picture? No. Okay. Interesting. I have another one of these discs as well. Somebody else have a disc in the back of the book? Okay, let's try yours. The one that I'm working with currently is Catherine's actually, and Afasa gave us one as well. But how well they work, I think, has to do with. What's that? We'll take it back. Okay. It might not even be the desk. I don't know. Things are getting boring. Let me draw some diagrams here for a minute. Okay, so when you start out with an oocyte, initially there's some stromal cells hanging around and some follicular cells. And they have some stromal cells. So what are stromal cells? Guys, you've heard about stromal cells before. Yeah, they're a support cell. Remember we said that we had this connective tissue. We could also call that stroma. So, connective tissue has fibroblasts in it. Stromal cell is, fibroblast-like cell. So they're calling this the primordial. So the primordial follicle, pretty simple. A couple of extra cells hanging around. Not that much complexity. And then they have the primary follicle. Once you get to primary, then you have the oocyte, but you've got more of these follicular cells forming a better barrier around the oocyte. And this is also where this membrane first appears. So what's kind of interesting about the zona pellucida? So we're just reviewing the maturation of the oocyte. But as we think about the maturation, as it gets closer to being the genuine item that's going to be fertilized, what additional characteristics does it need to take on? And one of the things is there's a growing group of cells around protecting this oocyte, not only necessary for its maturation, but some of those will be around even during that fertilization event. And one of those things that's around is the zona pellucida. So the zona pellucida makes these two proteins, ZP3 and 4, and they actually are attractants and receptors for sperm. And so they will attract and hold the sperm, and that's an important part of the fertilization of the egg. So therefore, I'm interested in the zona pellucida. I think that's something to know about. And then the other thing is that there's suddenly this transition too, and you deal with the next stage in the maturation of the oocyte. So you still have the zona pellucida, but now you've got so much more follicle here, a couple of layers thick. And that's where, instead of calling it follicular cells, we call this a granulosa. But it's the same cells. It's just more of them. But they're also taking on slightly different functions. So one of the things about the cells in the granulosa is that they're connected by gap junctions. And there's much more cell-cell contact than there was prior. So the follicle cells, the follicular cells, are not so interconnected, but the granulosa cells are. And then further diagrams of this, you get also in antrum and theca, and the theca interna and the theca externa. So I can draw those, but let's see if we can get this computer to go one more for us. Maybe I should restart the computer. Actually, the computer was on when I first started it. Maybe that's why I'm doing so badly. Maybe I should just shut it down. Okay, so the next stage beyond this was the vesicular follicle, and here it's a bigger mass. Still, you have the oocyte down here. It's off center. And there's some spaces in here. And this is what they call this antrum. So this is fluid filled. And the rest of this stuff in here, this is all going to be granulosa. So I think this is actually surprisingly significant for a simple diagram to understand what the stages are in the maturation of the ovary, and that the ovary is maturing as a whole follicle. And a follicle is just something that has lots of cells around it, and they start out being follicular cells, but then as it gets more complex, there's more and more things to identify. And one of the things that we identified is that the follicular cells are now called the granulosa cell, that there's this new membrane that surrounds the oocyte called the zona pellucida, and that eventually that gets even larger. And physically, I mean, in the book they show some sort of size difference, but even that size difference is not accurate. There's a much greater size difference. This whole follicle, as it's maturing, is getting bigger. Where is the follicle, Christian? Where is the follicle again? In the
ovary?
It's in the ovary. Yeah. So how many follicles are there in the ovary? 3? No, there's going to be lots and lots, right? So what's going to happen is that only a few of the follicles are going to start to mature. Sometimes it's up to about 20 follicles that can start to mature, but it seems like there's a competition or something. So some of the follicles are going to start to mature, but only one of them is going to make it all the way, or in some cases 2, but typically 1. And the 2 thing, one could happen, there could also be contribution from the other ovary, although there tends to be one dominant ovary versus, one of your ovaries is more active than the other ovary. It's kind of interesting. So then these surrounding cells, when you're forming this follicle, there's a couple things going on. One is that the oocyte itself is going to go through, it's stuck in prophase 1 for meiosis initially. And then it's going to, its cycle also is dependent upon how far it gets in terms of maturation. And ultimately, during fertilization, is the time where it actually resolves its final meiosis, which is meiosis 2. Okay, so it's letting me get to figure 1. Now let's see if I can get to figure 2. Yes. Okay, so here's the ovary, and here's these maturing follicles. And they're showing you how these follicles are changing over time. And we're saying that that most of them stay as primordial follicles, and during a cycle there's only a few that you want to bring through the further maturation steps such that they'll be ovulated. And we're saying that only one oocyte needs to be ovulated during one menstrual cycle, typically. And so, what happens to the other ones? Well, there's up to about 20 that start to mature, but then they don't mature further, and that fancy term was atresia. And so we're just looking at this. This is something that I think is very essential to the whole female reproductive thing. So remember, this is a histology class, and we're trying to put together, cells, understanding how cells and their morphology and their association with other cells into complex tissues, how that really describes what's going on inside that organ. So therefore, for this exam, this is a good thing to understand how the follicles are maturing, and that there's only a few that are going to make it all the way, and that maturation involves these additional cells. And the additional cells, one of the things that we were exposed to when we were thinking about the male reproduction system was that these support cells, or Sertoli cells, are, there's lots of those, and those are necessary. And then there's these interstitial cells, or Leyden cells, and those cells are producing testosterone. So same situation here. These support cells are going to be producing hormone that allow the oocyte to keep maturing. And that's kind of a crucial point. So, we were saying that initially these follicle cells, that when there's just a few, it's a follicle. Primary follicles have follicular cells, but once they start to go to the secondary follicle, there's more of them. You call it a granulosa, and then there's these this area in between, the antrum, which fills with fluid, and the fluid has got some of the hormone in there as well. So the hormone is what's it called again? It's like an androstenedione, androstenediol. And that's going to become estradiol. But it doesn't start out as estradiol initially. And that, again, molecules are, each time a molecule changes slightly, it gets a different name. And in biology, these names are given based on their biological function. But they're such related molecules, there could be very little that happens. In this case, I think there's one of the aromatic rings is clipped. An aromatic ring is that, like a phenol ring, the 6-member carbon thing. So if you break open a ring, then you have an aromatic, an aromanase enzyme will break open the aromatic ring. So that's that's something that happens where androstenediol is clipped and becomes estradiol, which then sounds familiar to people. Because I've talked to some people about this, and nobody remembers the androstenediol, but that's accumulating in the antrum and important for maturation. So the theca, internal and external. So let's look at this next one. This is what I was stumbling around trying to draw pictures of. And you see that, the sizes that they're showing you are not quite accurate. That this is, by the end, this is much larger than just the oocyte. But this theca interna and theca externa, these are cell layers that are producing hormone. Especially the theca interna is actually the one where the hormone is being produced. Theca externa is additional protection and support for that. So it reminds me of the Sertoli cell situation some, where it's an endocrine gland. It's actually, you're coexisting endocrine gland with the gamete that you're going to be producing, right? And you guys are familiar with that because in the male reproductive system, that was the way it was too. That there's endocrine cells, the Sertoli cells. The interstitial cells are an endocrine cell that's producing testosterone and allowing the maturation of the spermatid to being a fully functional sperm. So this is a similar situation here. So this Braphian follicle, that's just the name for the follicle that's actually going to be released. And, when this thing is released, some of these granulosa cells are surrounding the oocyte too, and they call that the corona radiata. Right, so you see that? And then even this base here, the base structure between some of these granulosa cells becomes the cumulus ophorus. And the ophorus, that's just a way to say, well, they have a special function because they're actually connecting these cells that are surrounding it. But the corona radiata is interesting because that's going to go with with this oocyte. When you have ovulation, you don't just get ovulation of the oocyte. The oocyte comes with a whole bunch of granulosa cells, the corona radiata. And I bet you guys wouldn't have thought of that one either. That's kind of a fine point, but it's not a fine point. Functionally, it actually turns out to be pretty significant. It's a way of protecting that cell, a way of protecting the egg cell. And then, but you're going to need to penetrate that too. How's the sperm going to get around this? So, I think that, yeah. I don't like to entirely bypass all the histological pictures because that's pretty important too. This is kind of cool looking, and there's an epithelia on the outside, but the epithelia is not very significant. But when people first started working with ovaries, they thought, well, there's this budding off that's occurring of the oocyte. So it must be these cells that are budding off, because we see them budding off from the surface. Isn't that funny? So then they gave them a special name that, that implied that they're actually generating the oocyte. But actually, as we know, the oocytes are in follicles. Here they are, and they're in follicles more complicated, and they have to go through this whole maturation, and eventually they're going to bud off, and they're going to come through the epithelia, but they have nothing to do with the epithelia. The epithelia, in this case, is a simple epithelia that's just covering the ovary. So this is a picture of the primordial follicle, and you see that these are really simple, right? Not a lot of support cells, but these are ovaries. And on the rest of the exams, one of the things I wanted to get away from was showing you pictures and asking you, well, what organ is that, right? Because that's not a real world skill. It was initially interesting to do that because I wanted you guys to emphasize similarities that you were seeing in some of the different systems. And we started out in the book, actually chapters like 1 through 4 are, giving you the basis of how tissues are designed and such. And so, to show you pictures and say, well, isn't this similar? Do you see some similar aspects? That made sense. But in the real world, if you get a biopsy from a patient, do you know where the biopsy came from? Oh yeah, you do. I mean, they tell you, this is a biopsy of, of an ovary, or this is a biopsy from this patient, and it comes from a certain area. So you know which organ you're looking at. So the idea of being able to distinguish liver from kidney from a reproductive organ is not so significant. So I'm more likely now on these tests to tell you what we're looking at a section of. But what I am going to ask you is, what's interesting about this picture? So what would you say about, this follicle here versus this one here, right? That's I think it's interesting. So you have the ovary and you have the maturation of the follicle containing an oocyte, and you see that the oocyte itself has gotten bigger, but it's got these support cells. It has the granulosa surrounding it, and so it's more mature than those guys. And this other picture over here, this actually shows you the zona pellucida, and it's a pretty dominant membrane. And it's just all glycoprotein, so there's no cells. But why are we so interested in that? Well, it's an extra attractant to sperm, and the sperm, the two proteins that are in here, CP3 and 4, they actually are like receptors for the sperm, and so the sperm bind to that. And what kind of picture is this? Catherine? All right, sorry. Catherine? You don't know? Rachel? What kind of picture is that? SEM, right? So it's a scanning electron micrograph. So of course you know that because it's black and white. You're looking at surface features, and this is the zona pellucida, and it's got this very ornate looking thing, but it's all protein, it's not cells. So if I asked you, what is that? Is that cells? You'd say, well, no, it's glycoprotein that's protecting the oocyte. Right? It's not cellular. But these are cells. And then what are those cells? Well, those are the granulosa. This is kind of a nice picture here of a mature follicle. You have the oocyte here. You can make out these different structures, including this atrium and its fluid-filled granular layers, and the theca interna and externa. And we were saying that what's interesting about the interna is it's producing the androstenediol, which is going to be metabolized into estradiol, which is going to be important for the maturation and release of that oocyte. Another similar picture is this guy. Just higher resolution of the same thing, where this is the granulosa theca interna, theca externa. And then what is A? Or an entropy. Okay, so it's time to look at this. This is where histology brings in genetic information. Sometimes we have examples of bringing in some of the molecular biology. But anyway, from the chromosome number, we know that we have 46 chromosomes and that they're pairs. So we have 23 pairs. And one of the 23 is a sex chromosome. Give me an example of a sex chromosome. Catherine? Okay, yeah, X and Y. Yeah, those are the two. So the 46 has X and Y, so it has 22. 44. And one is X. It's either XX or XY, and then on the 23, it could be there's 22 somatic chromosomes and one that's a sex chromosome, X or Y. And so when you start out with oogenesis, you're dealing with the complete diploid chromosome number, so you've got 46. And here's where we start to consider the follicle cells surrounding the primordial follicle. So the primordial follicle is very immature. It's existing within the ovary, and if you take a slice of ovary, you see mostly primordial oocytes. And that was the picture we looked at just moments ago. So there weren't a lot of granulosa cells around that. So during childhood, these cells are arrested in meiosis 1, which is, they actually haven't done they haven't gone through the division yet. So the chromosome number stays at 46. So if you look at an immature female and you were going to analyze the oocytes that are present there, they're all going to be immature oocytes, and they're going to be arrested in meiosis 1. How do you arrest in the cell cycle? How do you stop the cell cycle? You guys have all thought about the cell cycle, right? You have a DNA replication event, which is called the S phase. You have G1, G2 phases. Anyway, at the different phases of the cell cycle, there are these things called checkpoints. And you can stop the cell from going around the cell cycle and dividing its DNA and ultimately dividing the cell if you stop at a certain checkpoint. So there's a checkpoint here in early childhood where meiosis is actually stopped, and nothing happens with these cells until puberty. And then at that point, you're going to start to mature these oocytes, but you're going to mature them at a rate of about 20, they say approximately 20 primordial follicles are going to start to mature into the primary follicles every month. And that's where we were saying that, 20, yeah, that's a lot, but they don't really, none of them make it except for one. So they don't, they'll just atrophy and be reabsorbed. So, but some of them will continue. If they continue, then they'll go on. But okay, so primary follicle, and then there's the secondary follicle. The secondary follicle, that's where there has been there's only one secondary follicle that matures into the vesicular follicle. And that's where the secondary follicle oocyte is a haploid cell. So this is for the first time. See, everything up here is 46, and it's only when you get to the stage where you have a secondary oocyte that you have 23. And then, what is that corresponding to? This very mature follicle. The vesicular follicle is the follicle just before it actually is released during ovulation. That's the most mature one. So only the most mature one has the reduced chromosome number. That's kind of interesting, huh? So if we had asked you about that right at the beginning, what's the chromosome number in an oocyte, you would have told me 23. But it's not. It's not 23 up until that very moment where you're getting a very mature vesicular follicle that's about to be ovulated, and that's where the cell is allowed to reduce its chromosome number. What is this story with this first polar body? That also has 23 chromosomes. So polar bodies. You want the oocyte to be as robust as possible, and you want it to have as many resources as possible, because that oocyte is going to become, it's going to undergo multiple cell division, so it can become a very large cell. It's beneficial to have lots of cytoplasm, lots of machinery in there. The things that they discuss in the book every single time, which is, endoplasmic reticulum, Golgi, mitochondria, all that good stuff in the cytoplasm of the cells. You want the oocyte to have the benefit of all that richness. And so the polar body, you're going to get rid of this guy. You don't need this anymore. And so it has basically no cytoplasm with it. And so it's made to be discarded in favor of having this really robust secondary oocyte that can then go on and be released during ovulation. So one interesting thing is that there's a second meiosis. This is also not intuitive and not commonly understood. What is it that's actually being fertilized? And it's the ovum. The ovum is generated when the sperm is going to enter the oocyte. It's an opportunity for the female gamete to develop one step further and say, okay, we're going to separate out a second polar body. Another 23 chromosomes we're kicking out, and we've got this ovum, and that entity is what is going to combine with the 23 chromosomes that are present in the male sperm. It's kind of interesting, huh? Why would there be two things? But this second meiosis is only completed upon fertilization. Kind of shocking, and that's actually what's presented. It might be because maybe the nuclear membrane is breaking down, whatever, during a cell division, right? And so the chromosomes are going to be recombining with the sperm to make a diploid organism. And so how are you going to get it to do that? I remember when I was thinking about fertilization in high school, whatever, in biology class, and they said, well, there's 23 and 23 chromosomes that are going to get together. I was thinking, well, the sperm's got that flagella thing on it, so it's bringing in its 23 chromosomes, and it's just going to, force them in there. It's going to get all the way into the nucleus or whatever and force its 23 chromosomes in there. But that's not what's happening. The nuclear membrane needs to dissolve again, and you need to go through this other cell division, meiosis II, in order for those chromosomes to be available for recombining with the sperm's 23 chromosomes. It's kind of interesting. So what is this other thing? Corpus luteum. You guys know what that is? You ever heard of the corpus luteum?
It releases luteinizing hormone.
Okay. Why? Where's it come from? How is it made?
It's so once the oocyte is released, the ovary, or what's remaining from it, is a corpus luteum, and it kind of regulates the endometrium of the ovary of the uterine wall by releasing hormones.
Okay, so where does that make it? What kind of tissue is that? Corpus luteum.
Connective
See, on the test, I can have this column of, these different names, and then types of tissue that is. And so if I had corpus luteum, you would identify that as, you said it's producing hormones, so what kind of tissue does that make it?
Is it epithelial?
Yeah, it's epithelia. Epithelia are used highly because they're ducts, right? We didn't talk about ducts as much as we should have, but the topic has come up over and over again. Every time you have some kind of an opening, and you have the surrounding cells that are around the central opening, there's always it's always epithelial cells. So these have epithelial cells in them, but they're an endothelial function. So if I wanted you to match corpus luteum, I would say it's endothelial tissue because it's producing hormones, and the hormones are going to be critical to maintaining the wall of the uterus for fertilization. So it has an important role, but eventually it becomes a corpus albicans, which is just kind of a dead material. It's a bunch of collagen, and the cells die off and stuff, so that it becomes not unuseful. So why does it go away? It actually goes away somewhere in the middle of pregnancy. Well, at the beginning, it's useful when the female's first developing when the fertilized egg is first binds to the wall of the uterus, and it's going to be developing, and you create an amniotic sac. At the beginning, there's not that much hormone around, and so the corpus luteum is a way to have kind of a localized source of hormones. But once the support cells that are around the amniotic sac, once there's a greater supply of estrogen and progesterone, you don't need the corpus luteum anymore. You can kind of get rid of that. And so it just atrophies in the corpus albicans, and so it's not very interesting. So we had talked about this a little bit, but a lot of things begin with the pituitary. And it's the master endocrine gland, and it's going to be controlling ovulation to some extent. So there's the anterior pituitary and the posterior pituitary, but it all begins with the pituitary and the hypothalamus. And the hypothalamus is kind of a stems coming off of the brain. So the hypothalamus secretes gonadotrophin releasing hormone, which stimulates the anterior pituitary, and that is going to produce follicle stimulating hormone and luteinizing hormone that will stimulate follicular development. So there's the stuff that's going on inside the womb and all, but there's the brain and the hypothalamus, and then there's the pituitary in there, the master endocrine gland that's releasing these follicle stimulatory hormones and luteinizing hormone that's going to allow maturation of the follicles. And then, estrogen plays a key role. And estrogen is produced by the cells that are right around this maturing follicle. When it becomes a granulocyte, we had the theca interna. The theca interna is producing estrogen, low levels. The estradiol is a type of estrogen. Anyway, so there are low levels of estrogen that are required for the development of the follicle. And let's see, we go up here. There's a feedback loop to this, is what they're showing you. So then, once there's estrogen being produced down here with the fascicular follicle that's about to be ovulated, that there's enough estrogen there to feed back pituitary, and it's going to release luteinizing hormone, which actually then triggers ovulation. So it's only by your pituitary and the thymus, the hypothalamus is also getting the same signal. So it's a feedback loop, and it's affecting the levels of luteinizing hormone, which are going to lead to ovulation. And then once you ovulate, that's where you produce this corpus luteum that has an important role and keeps supplying estrogen, progesterone, and also inhibin, and those also feed back on the hypothalamus and pituitary. So all the parts work together, huh? It's kind of interesting. These are kind of ugly pictures, but I think they're ugly because they're not stained with any interesting colors. But this just shows corpus luteum. So, after ovulation, there's this remaining group of cells, and they look like kind of a bag of destroyed stuff. It's like after you take home the groceries and you still have the shopping bag, and it's kind of beaten up or whatever. Like that's what this tissue reminds me of. But it's still useful for keeping hormone levels going, so it's kept around a little bit longer. And then they show you kind of a higher magnification of what's going on here with granulosa lutein cells. They label them because they're it's important for them to keep producing luteinizing hormone. So it's really an endocrine type tissue at that point. Okay, so the rest of this stuff is, a lot of it is the structure of the other parts of female reproduction, and then also what happens during fertilization. So let's look at that. Major events of fertilization. So let's have the guys again tell us, see what they can recall. Major events in fertilization. So sperm is making its way up the vagina, and there's an egg there. So what has to happen? So already the story's gotten more complicated, right? The egg itself is not just the egg. There was a corona of cells around the outside of the egg. And then there's also the zona pellucida around the egg. So that sperm is going to have to go through a lot more than we thought it did. And then also, the egg is not ready to be fertilized, in a sense. I mean, it hasn't got through its final meiosis yet. And it's only triggered to go in its final meiosis when the sperm is actually connected to the egg. So already the story is much different than what we had thought it was before. But so tell me about what the sperm has to do.
First, make it to the cervix,
Okay.
into the fallopian tube.
Yeah. So how does it make it all? Is it just swimming? How is it making it all that way? Is it because, from the guy perspective, our sperm are great swimmers? Are the females helping out? Is their anatomy helping out there?
The cervix, there's a, is there a mucosa?
The what?
Is there a mucosa that has to dissolve before the sperm can enter?
Has to dissolve. Oh, there could be a plug.
Right.
But the plug is actually something that happens after pregnancy. So they don't need any more fertilization at a certain point, so they plug up the opening. It's kind of interesting. So, okay, so the male sperm is in there, and it's got to swim. It's got to make its way from the top of the cervix through the uterus into uterine tube.
Right.
It often ends up in the ampulla. which is just before the infibulum thing, and then the little finger things. So what's why does it have to swim all the way up there? Well, because ovulation, the oocyte doesn't come all the way down, right? So I mean, the sperm are better swimmers than the ovary, and the ovary has got this corona of cells around it as well. So so it's not going to go very far, but our sperm are there. So then the females they produce mucosa, but is it just simple mucosa or mucus? Or they're making mucus, but there's actually fluid there as well, and it's a nutritive to maintaining the health of the sperm. So how long? So a female ovulates, and then we were talking about this thing here. We were looking at 14 days. There was this period of 14 days. We weren't sure where the 14 days were, but they're certainly after the menstrual cycle, right after the menstrual cycle. So what about that ovulated ovary? The ovary ovulates, and then there's a release of an oocyte. So that's going to hang around for 14 days? No. So the oocyte, actually, it's only going to exist for a day. So actually, the female is fertile for 1 day out of her whole cycle. Now, why does it end up to be a little bit longer than that? Because the male sperm lasts a couple of days also. So if sperm are around, and ovulation occurs 2 or 3 days later, you're still good. So fertilization will occur. But it's actually, fertility is very short, surprisingly short, and it's not 14 days. But so what has to happen is the sperm need to deal with this corona radiata, and Connor, you had mentioned that process is
The acrosome producing the digestive enzymes.
yeah. Yeah, what kind of enzymes are those? What class of enzyme? It would break down lysozyme?
Would break down what? Protein?
Yeah, it would probably be a protease, right? So it actually turns out to be like a trypsin-like protease. Okay, so then there's also a hyaluronidase that's released by the sperm. And what is hyaluronidase? So what is hyaluron again? Or hyaluronic acid? Hyaluronic acid. Remember, that was that molecule that soaks up a lot of water, and all sorts of soft tissue have the need to retain water, and it's sort of spongy material. When you want to have cells being able to move through something, you want to have it be mostly water, so you produce hyaluronic acid or hyaluronin, and there's a lot of hyaluronin around this corona radiata, and so the sperm actually also are producing a hyaluronidase. So they're helping to break that down. Their objective is to make it through the corona radiata, right? You don't want the corona radiata to be an impenetrable barrier. I mean, you guys didn't even know that existed probably prior to our discussion, right? So, I mean, you were just thinking there's an oocyte there, right? But it's actually more than that. It's taking a whole bunch of its granulosa with it, and that's called the corona radiata once the cell has ovulated. And the sperm's got to make its way through that too. So the acrosomal reaction is going to be separating those cells. And how do you get through cells? Well, you produce a protease for one thing, that's always good. And then if it's kind of loose cellular structure, a hyaluronidase is good because hyaluron is a large molecule holding lots of water, and you just need to get all that water, spongy stuff out of the way. So you get that out of the way. And then you're dealing with just this coating around the oocyte. And that's the zona pellucida. And then remember that was kind of just this coating. It's not cellular, it's glycoproteins. And CP3 and 4 are on that coating. And then that's where the acrosomal reaction, the protease is called acrosin. And it digests the zona pellucida. Okay, so then what happens? So you get the first one in. John, what happens then? Well, I mean, just as a theoretical thing, without naming a particular thing, what do you think would be important? You've got one sperm is now made it to the egg, and that's probably as far as any of us have thought. We didn't know that there was a corona of cells around the oocyte to begin with. But so once we've gotten into there, and probably we didn't think too much about the zona pellucida either, and the fact that there's particular receptor proteins on there. But so once we've gotten to that point, though, then what else do you know about biology? Do you remember once the sperm gets in there, what does the egg have to do?
Divide.
That's what?
Divide.
Survive?
Divide. Divide. Yeah.
Yeah, it needs to divide, but is it all good? I mean, usually where there's one sperm, there's like a million, right? Isn't that?
So then when one sperm attaches itself to the egg, the other sperm can attach, right?
Yeah. So why is that? Why can't a second sperm attach? And why would you not want a second sperm to attach?
Because the sperm already has the chromosomes, and you don't want to mix with a different sperm.
Yeah. Yeah, it would mess things up. Yeah, always.
You wouldn't call it mutation, though.
Yeah, I think usually it ends up dying. You know, if there's more than one sperm that's gotten through. But the cell is set up to not allow that to happen. So there's a reaction that spreads like a wave across the entire surface of the oocyte, right? And that converts the zona pellucida to an impenetrable paravitelline barrier. So it's the same set of glycoproteins, but they are now called a different thing. So there's nothing, it's changed a little bit chemically, but it now presents itself as a barrier to sperm. So isn't that kind of interesting? It starts out as the receptor. Particular proteins, specific VP3 and 4, receptor proteins for sperm. They're inviting them in and everything, and then once one gets in, there's this wave that goes through this calcium-modulated ion flux that causes the whole zona pellucida to become now this gate, where it's like gating out the whole oocyte, and no further sperm can get in there. Okay, so then from the female side, what happens? So now the sperm is in there, so Catherine, what would happen next? Rachel, what would happen next? So is that oocyte ready to go? Is it ready to be fertilized? We had just talked about this. I mean, is it ready to recombine? Is its chromosomes ready for recombination with the male? Sperm is in there. This is late in the game. So fertilization has actually occurred. All those amazing steps and everything, it's all occurred. And sperm, that one very special sperm has gotten through, and it dumps its 23 chromosomes in there. Is the female side ready? Are you guys ready with the 23 chromosomes that you want? No. See, women are always late. Just could get in trouble with that. No. So you're not quite ready, right? So what else has to happen?
Second mitotic division.
The what?
Second mitotic division.
Yeah, second mitotic division, right? So there's a little bit of selection again. On choosing the 23 chromosomes that are going to be presented and are going to be recombining with the male. So there had been a block, a cell cycle block to meiosis 2 prior to that event. Kind of interesting. Okay, so then we have the fertilized egg, and it needs to. It was fertilized up here in the uterine tubule, and the uterine tubes are. It's actually the ampulla, which is sort of at the, very close to where the ovary is, and so it's got to make its way down the uterine tube into the uterus, and so. It can't swim anymore. It's not being propelled by the sperm, so it's going to be brushed along, right, with some cilia. It's got to be, and there's also going to be contractions. The uterine tube knows to subtly contract. What kind of cells are mediating those subtle contractions? You don't want a lot of force. You don't want skeletal muscle type force. Yeah, just smooth muscle, light contractions, a little bit of motion and some cilia beating, and that's enough to bring that fertilized egg down into the uterus. What happens if it doesn't get to the uterus? If it were not to get to the uterus and it was going to stay in the uterine tube?
Common pregnancy.
Yeah, topic pregnancy. How does that go?
I mean, implants on like the fallopian tube wall or anywhere around there.
Yeah,
It's bad news.
bad news. Yeah, so that's a disaster, right? So the chance of taking that to term is zero, pretty much pretty close to zero. Unless actually, I think in some cases they might be able to go in and coax it down.
Yeah.
Yeah, so there's some hope that doctors will be able to do something for you. Okay. So while we're talking about these other parts, so now we've gone away from the ovary. We're not discussing ovary anymore. We're talking about pregnancy. And so then there's the rest of the female reproductive system. And less interesting, but somewhat interesting. I don't know. I mean, I think, I will definitely emphasize the maturation of the ovary, of the follicle, and the ovulation events, and some fertilization events. But, you should know a little bit about this. So we were just saying that the uterine tube, if the pregnancy is taking place in the ampulla, then this fertilized egg's got to make its way down the uterine tube, and it's being brushed with cilia, and there's some subtle contractions, and you'd like it to implant somewhere on the wall of the uterus, on these walls. It could be, sometimes it's implanted even on the fundus, but mostly on the uterine walls. And what do these sections look like? So the uterine tube looks like this in a cross section. Why does a cross section look like that? Are these things floating in there? When you see a cross section that looks like that, is that floating in there? Afonso, what do you think? What explains that? You have a tissue slice, you look at it in the microscope, it looks like this. And you know it's from the uterine tube. What is this stuff floating? So there's some kind of lumen, right? The central part, that's not a word you can always use, is lumen. So there's a lumen here for the uterine tube, and it's filled with this stuff. And it's around the outside is a muscularis, so we were talking about the smooth muscle contraction, so that would explain that. But what is this stuff blocking the way of the lumen? That can't be good. Is that floating in there? Is that stuff floating in there? Tissue floating in there? It's what? Yeah, it's mucosa. So, part of it is an artifact of histological preparation. And remember that we brought that word up early on, that what you see, you always have to interpret what you see. You know what you did to it physically and then what you got. So if you take a thin section of something, if you have a folded piece of paper, like that, and you were going to slice through it, you could end up with something that looks different than the fact that it's actually all one smooth surface. So the fact that there's these little pieces, it's just convoluted. It's a convoluted mucosa, and it's a mucosa because you're very interested in having a lot of mucus there. Mucus is supportive and nutritive to the fertilized egg, and so the uterine tube should certainly have that kind of thing. But you take a section of it and you end up with these little pieces, and it's actually because there's a piece hanging off, and then you slice through it. It looks like it's just hanging there, but it's actually not. But oftentimes, too, remember the lumens are not just clear openings. Remember, even in some of the larger organs, when we looked at the lumen, there was a lot of material in there, a lot of tissue in there, and once there's fluid flowing through, greater, it can expand the lumen. But a lot of times the lumen is this, it's not just a circle. So you guys don't need to see that to say, oh, that's a lumen. Right? Sometimes a lumen, the actual clear part is only some part like that, and it's very convoluted or whatever. And in this case, in the uterine tube, there's all sorts of other stuff that looks like it's just hanging there. It's still a lumen. It reflects how convoluted the interior of that lumen is. And then, in a lot of these pictures, it's even hard for me to see any of these simple ciliated columnar epithelial cells, but the cilia are really hard to see. Okay, and then here's a section of the wall of the uterus. And you see, of course, it's going to have an epithelia. So the inside is coated with an epithelia, simple epithelia. And there's some uterine glands, basal layer. Cross section of the uterine wall of the ampulla. Again, it's an open lumen, but it's very convoluted epithelia in there. Interwoven layers of smooth muscle, and we've seen that before. That's very typical, right? So when you see something that looks like longer cells, and then something that looks like just little spots, and you see layers of them, that's always longitudinal versus perpendicular cell layers. And if you slice into it, the long ones going that way, and the ones that are at right angles to that, they look like just little spots. And you see the long ones and a bunch of little spots. So layers of muscular tissue to help guide the fertilized egg along its way. And then subsequent pictures, higher magnification, right? So low magnification here, higher, and even higher. And that's something that you guys could do for me, too, right? If I show you a low magnification picture and I say, well, what's the difference between this, this, and this? You say, well, this is the lowest magnification, you see the most cells, but it's, you see the morphology of the tissue overall, but as you get higher magnification, you can see the individual layers of cells. And if you really want to focus on particular cell types, as they do here, some of these cells, these are ciliated cells intermittent with these PEG cells that are producing whatever hormone or something. No, I guess mucosa kind of thing. So they're both helping the fertilized egg to get out, but they instead of having just all ciliated cells, these PEG cells are also useful in terms of adding fluid to allow this thing on its way. Okay, the endometrium. endometrium was until I was in college, I think. And I had heard, women talking about, that's part of part of this cycle. Well, there's this coating on the uterus that's going to be sloughed off during menstruation. So so the endometrium is that coating, and it's getting thicker, and it's getting thicker based on hormones. And so higher levels of estrogen and progesterone will trigger that. See, that's what they're showing here. So here's our cycle, Aryan cycle, and then here's levels of gonadotropins. And you see that they peak, follicle-stimulating hormone, luteinizing hormone are peaking just before ovulation. And then if you look at estrogen and progesterone, they have a different time course where they're, estrogen is peaking a little bit before and progesterone a little bit after. But then physically, inside the uterus, the lining of the uterus is changing in its thickness. So that's kind of a key point. And it's correlating. It's controlled with mostly estrogen and some progesterone. So that's why it's becoming the thickest at this point. So why would that make sense? Structure function. Why does the uterine lining need to get thicker post ovulation? Elizabeth, what do you think? Why does this have to happen? Why does the uterine lining have to get thicker following ovulation? It's getting so much thicker. And then all of a sudden it drops off. It gets very thin again. And then it needs to grow back up. Yeah. So a fertilized egg, it wants to land on a very rich bed of tissue that's really well vascularized. And so having the lining at its thickest point, where just after ovulation, makes a lot of sense. And then why do you want to slough off the whole lining once that's not going to happen? It's pretty wasteful. Seems wasteful. It's a lot of work. But, is it worth it? Reproduction for biology is what it's all about, right? So that, the body's willing to pay high prices to reproduce and to guarantee the next generation. So reproducing this, resynthesizing the whole endometrial layer is worthwhile. So they're all fresh vessels. There's no bacterial contamination. There's nothing, it's all fresh tissue, well vascularized. It's worth going through that every time you present this valuable egg. You want to have that possibility there, and that's controlled by estrogen and progesterone. What happens in the environment if remember you guys have heard about BPE, like in the plastic bottles that you're drinking water out of? There's a plasticizer that keeps the plastic soft. And it's bisphenol A, BPA, I guess. Bisphenol A, have you heard about that? Bisphenol A has a structure that's very similar to estrogen. So if your body is exposed to higher estrogen, that would have, potentially could have the effect of causing this lining to get thicker, even thicker. So then menstruation there might be a thicker layer to menstruate from drinking out of your plastic water bottle. Kind of a frightening thought. But anyway, this is kind of a current issue in society right now, thinking about our use of organics. The petroleum industry is digging up, petroleum and we're making different things out of it, and kind of anything goes right now. Anything that we can make, we do. And then sort of later on, we think, oh, well, what about this type of plastic? What about the type of plasticizer that's in here? What about toxicity of melamines, an additive for plastics? Some of these things are carcinogenic. Some of them are hormone analogs. And so there's potential downside to that. So there's a condition called endometriosis where the endometrial tissue can grow beyond, they grow into the ovaries, oviducts, and elsewhere. So rather than just lining the uterus, this tissue kind of gets loose and keeps growing and goes places that it shouldn't go, and actually can even make it all the way into ovaries and oviducts and other places. So then that would have, that seems like it might be more likely to occur too if there were environmental contaminants that were analogs of estrogen or progesterone.
I feel like nowadays everything can cause like breast cancer.
Everything causes cancer?
Yeah.
So why bother? Why worry about it?
That's just my feelings.
I don't know. I'm still thinking it's worth trying to change things. For example, when I was in high school, everything was contaminated with DDT, and even the cereal I remember eating some shredded wheat in the morning, and I was listening to the radio, and they said, well, the government has now to announce the highest levels in consumer products that we're exposed to. And number one on the list was shredded wheat. I was sitting with my bowl of shredded wheat, shoveling it in, thinking, well, this is the highest level that's being offered to the public right now. So then I stopped. I threw away the box of shredded wheat at that point. So I mean, it's always like that. There's always a wave of things that are adopted by the society. It's an industrialized society, so we get into mass production of things. And then at some point we figure out, oh, that's maybe not a good thing. So then we try to backtrack and put some regulations in place. There was this latest thing on the trans fat thing. Did you hear about the trans fat? So they basically outlawed trans fats now. Or they're completely banned by 2016, but there's like a partial ban now or whatever. You're supposed to show that you're downsizing, down regulating the amount of trans fats in any kind of product. And it's because they don't metabolize correctly. In chemistry, there's cis and trans molecules, and if you go through, anyway, I don't want to get into that. You go into the reaction, there's both stereoisomers, and so the body can't really handle it that well. Okay, so just to finish up on this endometrial layer, how important that is. What does that look like? Cartoon. Pretty complicated. Myometrium, molecular artery. So there's lots of structures here. This spiral artery is kind of of interest because it's a very rich blood supply right at that point. And then this is where you have that endometrium. Uterine glands are going out. Wow, I'm always impressed how much there is here. So this shows some different phases in the uterus. And that's what I was saying, in biology, even if you have the same thing, as it moves through different phases, it's called different things. And so then you think, oh, there's this other thing that I don't know. But actually, you do know it, it's just morphing and changing. And it's going through a cycle. So the uterine cycle has different stages to it, and some of those are shown here. they're calling this a functional layer because it's close to the lumen. And in all these diagrams, L is always the lumen. If I show you any pictures and they have letters on them, you know from the last test that that's a give me, pretty much, because that's the first letter of the word that describes what that is. So if you see clear area and there's L, that's even more obvious that that's a lumen. F for a functional area, I'm not so sure, but that's not much of a giveaway. Usually F would be fibroblasts. Myometrium, sort of a more, at the bottom, a more muscular layer. Okay, we don't really need to look at that in more detail. Let's see. It's over here. So just to make sure that we have the steps understood on implanting this fertilized egg. So we've dealt with everything up to that point, but it's worth spending a few more moments just looking at this. So this open area here is a lumen also, right? Any opening. Lumen of the uterus. And the oocyte, the fertilized egg, has now become a blastocyst. How did it do that? What is a blastocyst? It's not a new thing, it's the old thing, right? It's just got through some more cell divisions. What would you, just looking at it, what's the characteristic of that? What does that look like? What shape is it?
Huge ball of cells.
Yeah, it's a huge ball of cells. So blastocyst is just, when you've gotten cell division, the fertilized egg is just a big ball of cells. And you're looking at the numbers of days from fertilization. And this has got to implant itself on the endometrium. And the health of the endometrium, as we had just gone over, is determined by hormone levels, and it's at its maximum thickness and maximum health at the point where the fertilized egg is coming down. And as it's implanting, you see that the egg is also maturing from a blastocyst to trophoblast, which has this embryoblast area in it. So there's this thing called the inner cell mass. And this is right in the early stages of implantation of this fertilized egg, where the inner cell mass is actually going to be the embryo. And then there's other associated cells, because the blastocyst is not only making the embryo, but it's got to make the whole amniotic sac. It's got the cells for the chorion. It's carrying that along with it. So this is going to become the embryo, and then it's got this other material that's going to become the chorion, the contribution of the egg to creating a sac. So then there's a couple of different stages to this. But basically it actually kind of gets right into the tissue with the cytotrophoblast. So I think that that's enough. I mean, I pretty much want you to know that it implants in the endometrium, and that it starts out as a blastocyst, and the fertilized egg is also maturing, and there's a difference between inner cell mass cells and the rest of the what's going to become a yolk sac here. So the contribution to the amniotic sac, the amnion here. So then this is the cartoon continued. So the endometrium has thickened, and this is the area where the implantation has occurred, and the placenta has developed, and you've got an umbilical cord that's connected to the embryo. There's some names for these layers: decidua capillaris and periolitalis, layers of this of the placenta. And then we had mentioned the cervical mucus plug. So that's we were saying the cervix is off limits once there's fertilization. It makes sense for no further. You don't want any further fertilization event to occur, and so there's a specific plug of mucus that forms there. So so it's it's okay to have further sexual activity because there's a plug there. You're not going to damage the developing fetus because it's plugged off very specifically. Okay. And of course, blood supply and the interaction between the maternal blood supply and the umbilical artery that's going to the placenta. So there's a kind of a intermingling of blood supply, trying to get the optimal. What else was there? This is the placenta containing chorionic villi. There's also some fetal blood there. Changes in the cervix. Pap smear. You guys know about pap smear? So cervical cancer is pretty dangerous, and it tends to be caused by HPV, human papillomavirus, and there's lots of serotypes of human papillomavirus. I've actually done some work on that before. There was close to 50, I think, different serotypes. So relatives of human papillomavirus whose genomes are slightly different, therefore their surface antigens are going to be a little bit different. So in other words, it's going to be difficult to make an anti-serum against HPV. Still, that's now available. So you can be immunized against HPV. So that's now being offered to people as a possible way to avoid getting exposed to a pathogen that could cause cervical cancer. It's made by Merck. The first one released anyway was made by Merck. So it basically immunizes you against the antigens that are the most common on the surface proteins of the most common serotypes of viruses that tend to cause cervical cancer. So out of those 50 different serotype varieties of the virus, they picked out the ones that are most often involved with cervical cancer, and then they put those antigens together into one immune shot, one injection, and then that will give some protection against HPV that could result in cervical cancer. So then there was this doctor in the 1920s that said, well, we can screen for cervical cancer just by scraping some cells off the surface of the uterus, and then we create a slide. And you guys have created slides in this lab. You just take a slide just like what we have, and we wouldn't ask anybody in this class to scrape their cervix, but they go in with just a little scraping, a little plastic thing, and they scrape off some cells and put it on a slide, and then stain it, which is just basically H&E stain, although he has orange G also in there, but it's hematoxylin, orange G, and eosin. So it's basically our staining kit back there, and then that gives you a slide that looks like this. And then what you're going to be looking for are cells that look cancerous. And what does a cancerous cell look like? well, they grow rapidly, and so they can be multinucleated. There can be they're more likely to be seen in division. Um, so I'm not an expert in that, but anyway, that ends up being a Pap smear. So you guys have heard about Pap smear. So that was just the sort of standard of therapy, and it's routinely suggested that women get regular Pap smears to assess the level of possible risk for cervical cancer. But then if you've gotten the immunization against HPV, then your chances are probably better. I think that vaccine is done well. I haven't read any reports about side effects from that, and it does seem to be offering some level of protection. Could you count on that being complete protection? No, because they arbitrarily picked out a couple of serotypes that were the most active, but you could end up with a different serotype that's a little more rare to cause cervical cancer. Still end up with cervical cancer in the end, but it just reduces your chances, and I think it's a nice advance.
Is that immunization only offered to females?
Only offered to who?
Females?
Because
my pediatrician offered it to me too. I don't know if he was just jipping me for money or.
yeah, I guess not. I guess that would work. Sure, why not? Yeah, because the males are carrying it too. I mean, females are getting HPV infections from males. I mean, that's the great thing about having two sexes. Like we cause trouble for each other on all sorts of levels. But yeah, so if there's an infectious agent, you get it from from sexual activity, and so yeah, I think it would work on a male. Why not? The thing about males, it's more visible too. So since we have an exterior sexual organ, you can look at it, and if you see something on there, then you're more likely to say, oh, what the heck is this? And then you go to your doctor, and there's more you can do about it also because it's on the exterior, so it could be, maybe cauterized or something like that. I don't know. Practically speaking, I'm not sure what you can do, but, I mean, at least you're aware of what's going on. Whereas the female, since she's got an internal sexual organ, then she can't look. She doesn't know what's going on. She doesn't know if she's been exposed to something. So it's actually, it's more of a male's responsibility to keep track of things because it's obvious and make sure that you don't do any damage there. So here's a picture of the vagina, and so pretty simple stuff. LP, lamina propria, which is we've seen in several organs, and a nice thick epithelia and the mucosal layer. So that's kind of what you'd expect. Over here, does this look like skin? Well, no. It's stratified though. What's similar about it? Well, these are flattened cells more at the top, and then you see that they change in shape. So that's a typical stratified epithelia. Nice thick epithelia, and then the cells look different as you're looking through the epithelia. Lamina propria tends to be the connective tissue where you have more cells and you don't want to create just a connective tissue. You have more ducts and more fluid and everything. You still have the basement membrane, so you see the cells kind of lining up along that interface between lamina propria and the vaginal epithelia. Okay, so the last thing is is breast. Has anyone here had a class with Dr. Louis? Yeah, most people. Yeah. What class was that? Biochemistry. So So she's on sabbatical right now, I think. So the current group doesn't have her. She was interested in what's causing breast cancer. But anyway, so the reason why this is lumped in with female reproduction is because it's triggered by hormones, and it's also providing the nourishment for the newborn baby, so it kind of makes sense to put it here. One of the things that we've gone through a lot of material, and at a risk of having too much stuff to deal with, at least you've gone through the whole body by going through the whole book, and I think that's a good thing, because in the past they've used this book and they've left out chapters in kind of I thought sort of somewhat random way. So it's better to have gone through the whole body, I think. And it's useful for exams. You never know what you're going to be asked about. So looking at breasts, there's a couple of terms that are kind of cool. Colostrum is a fluid that accumulates in ducts. So these are all ducts. So ducts are, what kind of cells are in a duct? Epithelial cells. Epithelial cells line a duct. And the duct is making something that's going to be secreted, and so you're going to want to encourage that secretion to go out. And so you typically have some sort of smooth muscle layer, some sort of subtle contractions that occur. As we were saying about biology, because things are changing, even if you have the same thing at one point, it changes at different points. So this network of ducts, even if it's mature but inactive, it has a fairly simple structure. During early pregnancy, this network is going to become much more complicated. Mid-pregnancy, it's going to start to fill with some fluid, and this colostrum fluid is the first thing that you're going to start to stockpile some nutrients for what's going to be eventually in the milk. You don't need the milk yet, though, in mid-pregnancy. But once you're actually begin lactating, when the child's been born, then these ducts are going to fill with milk. And so you're going to make use of colostrum fluid that had been building up. And you also want, since it's a duct and there's some biological control to that, you want the milk to squirt out at times where it's wanted. So then you create some nerve endings on the end of the nipple such that if those are stimulated, there's going to be some hormonal response that is going to encourage the release of milk. And so that's lactation. And there'll be a period of several months when a female is lactating and able to give milk. And then once that need goes away, then this whole network can regress and simplify down to where it was before, when it was inactive, mature but inactive. So the network is simplified out, and you don't have the fluid in those ducts. So what's in this fluid? Well, there's some proteins, there's some lipids, there's a lot of sugar. What kind of sugar is in milk? Lactose. So it's mostly sugar. Human milk contains over 7 grams of lactose per deciliter, which I guess is 10 liters.